The green algae are a large, informal grouping of algae consisting of the Chlorophyte and Charophyte algae, which are now placed in separate divisions. The land plants or embryophytes are thought to have emerged from the charophytes. As the embryophytes are not algae, and are therefore excluded, green algae are a paraphyletic group. However, the clade that includes both green algae and embryophytes is monophyletic and is referred to as the clade Viridiplantae and as the kingdom Plantae. The green algae include unicellular and colonial flagellates, most with 2 flagella per cell, as well as various colonial, coccoid and filamentous forms, and macroscopic, multicellular seaweeds. In the Carolus, the closest relatives of higher plants, full cellular differentiation of tissues occurs. There are about 8,000 species of green algae. Many species live most of their lives as single cells, while other species form coenobia, long filaments, or highly differentiated macroscopic seaweeds. A few other organisms rely on green algae to conduct photosynthesis for them. The chloroplasts in euglenids and chlorarachniophytes were acquired from ungested green algae, and in the latter retain a nucleomorph. Green algae are also found symbiotically in the ciliate paramecium, and in Hydroviridis and in flatworms. Some species of green algae, particularly of genera Trabalcaceae of the class Trabucciaceae and Trentepolia, can be found in symbiotic associations with fungi to form lichens. In general, the fungal species that partner in lichens cannot live on their own, while the algal species is often found living in nature without the fungus. Trentepolia is a filamentous green alga that can live independently on humid soil, rocks or tree bark, or form the photosymbiont in lichens of the family Graphidaceae. Cellular structure. Green algae have chloroplasts that contain chlorophyll A and B, giving them a bright green color, as well as the accessory pigments beta-carotene and xanthophylls in stacked thylakoids. The cell walls of green algae usually contain cellulose, and they store carbohydrate in the form of starch. All green algae have mitochondria with flat crista. When present, paired flagella are used to move the cell. They are anchored by a cross-shaped system of microtubules and fibrous strands. Flagella are only present in the motile male gametes of charophytes and are absent from the gametes of pinophyta and flowering plants. Members of the Chlorophyceae class undergo closed mitosis in the most common form of cell division among the green algae, which occurs via a phycoplast. Like land plants, charophyte green algae undergo open mitosis without centrioles. Instead, a raft of microtubules, the phragmoplast, is formed from the mitotic spindle, and cell division involves the use of this phragmoplast in the production of a cell plate. Origins. Photosynthetic eukaryotes originated following a primary endosymbiotic event, where a heterotrophic eukaryotic cell engulfed a photosynthetic cyanobacterium-like prokaryote that became stably integrated and eventually evolved into a membrane-bound organelle, the plastid. This primary endosymbiosis event gave rise to 3 autotrophic clades with primary plastids: the green plants, the red algae and the glaucophytes. Evolution and classification. Green algae are often classified with their embryophyte descendants in the green plant clade Viridiplantae. Viridiplantae, together with red algae and glaucophyte algae, form the supergroup Primoplantae, also known as Archaeplastida or Plantae sensulato. The Viridiplantae diverged into 2 clades. The Chlorophyta include the early diverging Prasinophyte lineages and the core Chlorophyta, which contain the majority of described species of green algae. The Streptophyta include charophytes and land plants. Below is a consensus reconstruction of green algal relationships, mainly based on molecular data. The algae of this paraphyletic group, Charophyta, were included before in Chlorophyta, so green algae and Chlorophyta in this definition were synonyms. Reproduction. Green algae are eukaryotic organisms that follow a reproduction cycle called alternation of generations. Reproduction varies from fusion of identical cells to fertilization of a large non-motile cell by a smaller motile one. However, these traits show some variation, most notably among the basal green algae called prasinophytes. Haploid algal cells can fuse with other haploid cells to form diploid zygotes. When filamentous algae do this, they form bridges between cells and leave empty cell walls behind that can be easily distinguished under the light microscope. This process is called conjugation and occurs, for example, in Spirogyra. The species of ulva are reproductively isomorphic. The diploid vegetative phase is the site of meiosis and releases haploid zoospores, which germinate and grow, producing a haploid phase alternating with the vegetative phase. Sex pheromone production is likely a common feature of green algae, although only studied in detail in a few model organisms. Volvox is a genus of chlorophytes. Different species form spherical colonies of up to 50,000 cells. One well-studied species, Volvox carteri, occupies temporary pools of water that tend to dry out in the heat of late summer. As their environment dries out, asexual V. carteri quickly die. However, they are able to escape death by switching, shortly before drying is complete, to the sexual phase of their life cycle that leads to production of dormant desiccation-resistant zygotes. Sexual development is initiated by a glycoprotein pheromone. This pheromone is one of the most potent known biological effector molecules. It can trigger sexual development at concentrations as low as 10-16 m. Kirk showed that sex-inducing pheromone production can be triggered experimentally in somatic cells by heat shock. Thus, heat shock may be a condition that ordinarily triggers sex-inducing pheromone in nature. The Clostridium pericerosum strigosum littoral complex is a unicellular, isogamous charophysian alga group that is the closest unicellular relative to land plants. Heterophilic strains of different mating type can conjugate to form zygospores. Sex pheromones termed protoplast release-inducing proteins produced by mating type and mating type cells facilitate this process. Chemistry. The green algae span a wide range of delta-13C values, with different groups having different typical ranges. Green algae contain sulfated polysaccharides like remnant sulfate in the amorphous sections of their cell walls. Some species also contain gallotannins. Physiology. The green algae, including the Charophytes algae, have served as model experimental organisms to understand the mechanisms of the ionic and water permeability of membranes, osmoregulation, turgor regulation, salt tolerance, cytoplasmic streaming, and the generation of action potentials.